Yeah. So thank you, Birgit, and also for the kind invitation and having me in this webinar today. So as Birgit mentioned, my name is Andrea Croselli-Strehle. I'm the head of the core facility Computational Bioanalytics at the Medical University Graz. And today we'll talk together with my colleague Michael Gruber about OMERO. And he will also give you a live demo. I think this is the most interesting thing of today. And on the next slide, I just want to give you a short impression about our data management ecosystem. So you can see we have different platforms. Most of them are open source tools, and all those tools are connected. So you can see, for example, RedCap, where we collect and manage clinical data. This data can be also connected, for example, with bioimages. So assume that maybe you're working on a breast cancer cohort, you have clinical data, but maybe you have also whole slide images. Then you store them in OMERO. And maybe additionally, you're interested in proteomics, metabolomics, spatial transcriptomics, whatever. So for all the omics data, we use IRODS. Nevertheless, next to the clinical data, maybe you also have specimen, and you also can connect your clinical data to the specimen and or the pre-analytical data that give you information about the quality of the specimen. And all those informations are stored in a tool. This is called Open Specimen. And on the right side of this slide, you can see that we use ELAB FTW, where you can document all the work you do in the lab. And the last tool, the Pirate system, is then relevant if you're working in the context of animal experiments. Additionally, we have HPC cluster called MedBioNode. And this cluster is connected to the mentioned platforms. Currently, we have about 300 researchers that daily use the MedBioNode cluster. And on the top of this slide, you can see OpenSearch. This is our research data discovery tool. So this means whenever you store your data, you can search them within the specific tools, but also in the Open Search tool. But there, the Open Search tool is, I would say, more for researchers that who want to find out, are data available maybe for secondary use of research? So assume you have your research question. You want to find out, is it necessary to go into a prospective study, or are data available? And therefore, it's needed that you have your data management systems, that data are connected and searchable and findable. I don't know where you are coming from. And I also don't know if you have such a data management ecosystem at your research institution or not. So if you would start from the scratch, I would recommend to find out what exists currently, and to define what are specific requirements from your researchers, from local stakeholders, but also from national initiatives. So I would not replace tools. We go more in the direction of integrating additional tools if something is missing. And we have a strong focus on use cases as a compass. As I mentioned, this example with a breast cancer cohort, this was also a chance for us to find out which services are really needed along the entire data lifecycle. So this means you need, on the one hand, data management tools, and on the other hand, services. So this is a really important thing, not just to deploy a tool and say, this is it. Michael can give you some impressions about his daily work that is really important to offer services like consultations, trainings, e-learning tools, but also things like automized data transfer pipelines. So this situation, maybe we can explain a bit. You have a device where you collect your images, and then on the next step, you want to transfer the data to OMERO. And this is a critical part. And if you want to support your researcher, you will go into automation. Maybe Michi can talk later a bit more about this. And one additional important point are the metadata to the images itself. So we all know the standard garbage in, garbage out, right? So why do we need metadata? If you only store the raw data in OMERO, then it can be tricky to find them. And also for secondary use of data, we need the metadata to give the information to other research groups which data sets are already there. So here, this is such a workflow where you can see we have here a device for image acquisition, and we need a pipeline that gives us the possibility to transfer the images to OMERO. And from OMERO, you have different possibilities to analyze your data. You can do some parts of analysis in OMERO itself. You can connect OMERO with other tools like Imaris, or you can analyze your data on the MedBioNode cluster itself. And this is just an example for metadata by design. So what does this mean? You can imagine if you give a researcher a template, yeah, and say, okay, these are the data, metadata we need, please fill this out. Then I would, as a researcher, open the file and close it within a second, right? Because there is so much information there, and I would not be sure if I'm able to give all this information. Therefore, we say, okay, we need data stewards. They support this part, and we prepared examples for metadata in different specific fields. And this is a bit of the way we work here at the Medical University of Graz, not just to deploy tools, also to define dedicated services and offer both together. And now I want to hand over to Michael Gruber. He is our expert for OMERO. Michi.
Thank you, Andrea, for this kind introduction. My name is Michael Gruber, and I'm working on the topic of OMERO for the last 1.5 years. And in my part of the presentation, I'm going to give you an overview of OMERO and a short introduction. And I also want to add a short remark that this presentation is full with many links, which you may find useful if you want to dive deeper in this topic, OMERO. So let's start with the question, what is OMERO? And to answer it, OMERO is an open source platform for managing image data, especially microscope image data. And it was developed by the Open Microscopy Environment Consortium, short OME, and provides a centralized environment based on a client-server architecture where the images are accessed over the network by clients. And it isn't only a repository, but it is a whole environment which supports storing, managing, visualization and analysis of microscope images and the associated metadata. So in short, OMERO stands for Open, according to open source platform, microscopy, the type of image data, environment for the whole platform, remote for the way of access, and objects, the type of images and metadata. OMERO consists of 4 main components. The most important part is the OMERO server. It is used or takes part. It does all the calculation in the background and communicates with the other components. The component the users see is OMERO Web, which is the client, which can be used to access the image data and manage it. There is also a database which holds the information about all the user and group data and establishes the links, holds the information data, attaches all the annotations, links, and so on. And the 4th part is the attached network drive, which is actually used to store the image data. So why should one use OMERO instead of a different software for handling images? So one of the main reasons is it is open source. And it is not just open source, but actually also well documented, which is not often the case for open source software. It comes with a strong community. So there is still a lot of development ongoing and driven by the community. It is widely used across the German-speaking countries. A lot of universities use it there and institutions. And there are frequently workshops and meetings taking place with a very nice community, which is very helpful and supportive. OMERO supports a large number of bioimage formats, more than 140 different image formats. I want to open the website. I hope you can see this. And this gives you an overview of the supported image formats. So there are a lot of supported formats from different vendors and different microscopes, technologies. And yeah, so however, the different formats are supported differently. In one are supported very vastly, and some are very supported only in a minor form. As I said before, it concentrates mostly on microscope formats, but however, there are also different forms supported, like DICOM, for example, and open image formats as well as OMETIF. Yeah. So let's go back to the presentation. During the import of the data, OMERO keeps the original file format and does the conversion on the fly during viewing the data. Also, during the import of the images, the metadata, the technical metadata, which is attached by the microscope, is captured and preserved. And further, it is possible to attach a lot of different forms of metadata to the images and other OMERO objects, which helps to preserve the experimental contexts and helps to regard to the principles, to the FAIR principles. It enables the controlled sharing of data. We have selected users in so-called research groups. It offers a centralized data environment, and which also helps to streamline workflows, because one platform is used and therefore the same script can be used for various tasks. And it comes also with a lot of different APIs and plugins, which I will consider a little later in my talk. So Andrea previously told a little bit about how OMERO is integrated into the system of the Med Uni Graz. And here I want to give a short demonstration how the various components of OMERO include into this system. Here you have the backbone, the server, the OMERO server with the database and the attached network storage. And over the network, various clients can communicate then with the server. For example, microscopes are attached, and also importer software is used to upload the images into the OMERO environment. Then various clients can be used, especially the OMERO web client. There's also a connection to the HPC cluster of the university, which can be useful to analyze your image data. And there's also a vast amount of plugins, like plugin for QPath, Cell Profiler, and Fiji, which are vastly used for image analysis, as well as APIs, especially for R and Python. Python is supported with an official API, and it also comes with the so-called OMERO API, which helps to break down various programming tasks into easy functions. So, in order to set up OMERO on this setup an OMERO server, there's just a minimum of hardware required to run it for approximately 100 users, where around 10 users are working at the same time. There's also an optimum configuration pronounced or hosted on the official OMERO website, which they say is used to be secure for the next 4 years. And here at the Med Uni Graz, we use the following configuration. We use a CPU with 12 cores and 32 gigabytes of RAM. The system runs in a virtual machine, in a Docker system, on a Linux OS, and the system is backed on a daily basis by taking snapshots of the virtual machine, and the images are included or uploaded to an attached 200 terabyte network drive, which is included into the virtual machine or mounted. Yeah, and the images are uploaded over a network. So this system, like we have here, has the advantage that with this attached network drive, it is scalable, and the images are backed up on a daily basis as well. The setup of OMERO can be done either natively, directly on the host system, or by using Docker images. And the setup with the Docker images is very simple, so I want to go through the several steps in order to show you how simple it is. You just have to set up a Linux server, and then install the Docker on the OS. Go here, use the following link to go to the download the Docker Compose file. Open this file in a text editor and change, just exchange the marked names, passwords, and so on. Then open a bash shell, navigate to the directory where the .yaml file is contained, and then run just the command docker compose pull, where then Docker starts to download all the Docker images and to start. And then you can start the Docker compose up command, and which then creates the containers and runs OMERO. And all there's left is then to open a browser and to navigate to the URL which was specified and log in. OMERO also supports the single sign-on login. And if you do this, the OMERO web window will open with showing your user repository containing all the projects which you have already created and your data which you have uploaded. To give you just a short overview how OMERO Web is composed, you have here the main menu, which helps you to navigate through the main parts of OMERO. It is here possible to switch your repository from your user repository to group repositories you are a member of. You have here a certain management tools which help you to manage your repository. On the right side, you have here the so-called info field, where important information about the objects here in OMERO are viewed. You have here a user menu which you can open, which gives you information about your account. And also here a field for metadata, which you can add to the images or other objects. You have here the images, which are shown as thumbnails. And here is a button to open and run scripts, various scripts on the images, which are part of this data set. There are two types of image data in OMERO, so-called normal image data and so-called high content screening data. Normal image data are just plain images, when high content screening data are created by plate screening. If you are used to structure your data via normal Explorer directories, where you can put a subfolder and a subfolder into your project in order to organize it, and where the project or the folder names contain the information, you have to rethink this process a little bit if you are using OMERO, because OMERO only comes with two hierarchy levels for each, for both data structures. You have for the normal data structure, so-called project level, dataset level, and then already there are the images. And for the high content screening data, you have the screen and the plate folder, and there are already then the various runs. Okay. So this is a short example how an repository can look like. OMERO, there are several objects in OMERO which can be uploaded or defined or created. The so-called regular OMERO objects are project folders, dataset folders, images, screens, and plates, whereby there is a separation of so-called parent objects and child objects, where a parent object can contain child objects. If objects are created or images are uploaded, then there is an ownership assigned to this object, which is unique. The owner is always the one who is logged in and creates the object. However, this can be changed afterwards. Also, if an object is created or an image is uploaded, there is a unique ID assigned to the object, which cannot be changed and which is used in order to manage all the data in OMERO. And additionally, there's also an object name, which is, however, user-defined and not unique and can be changed every time. Now some words about how data is organized in OMERO. The data is organized by so-called link-based model, where the data is not duplicated, but for example, if you copy and paste the data, just links are created, and where the links are then can point to several different locations, which helps to keep the amount of used space, memory space, at a minimum, and to prevent the duplication of the data. However, if objects are deleted, then the objects themselves are deleted, and not only the links. Okay, so how do I get my data into, or my images into OMERO? There are several different ways, like for example, via an API, which helps to import a vast amount of data. But the most used way is to use the OMERO Insight desktop client, which is installed locally on a PC. And once started, shows an overview of the repository, same as OMERO Web does, and has also then the option to import data, which when I press this button, the so-called OMERO importer window starts, where on the left, the local directories on my PC are shown, and on the right side, there is the already loaded or created import queue shown. So if I select one or several images and then press this add to the queue button, I create then a data import queue to upload the images. However, there is due to the two data types, various different data types, there is two ways to import the data, either through projects to import, for example, slide data, or through screens in order to import then the high content screening data. Here you can see how high content screening data looks like. There is, for example, a 96 well plate containing holes where several tissue samples are inside of these holes, and then, for example, various treatments are then done on this part, and the plate is then put into a plate reader, and all the data is read out at once. So after I have an import queue, I can then there's the option to define various import options, for example, or add another adds useful information which is not included yet. For example, old microscopes do not add how much micrometer 1 pixel is into their information. So this type of information can be installed, can be defined here afterwards, or there's also the possibility to skip various calculations then during the import process. During the import process, not only the pictures are uploaded to the OMERO server and OMERO platform, but there are also several calculations done, like so-called pyramidization. This helps to fast view your data, even big image data, over the browser, over the local network, or even over the internet everywhere on the world, where you have access to your data. And so if you have your image uploaded here, then you have it with a certain magnification, and OMERO then calculates also thumbnails of various different magnification levels, like when you zoom into the world map in Google Street View. The success of your import is then shown here via these green check marks. And after you refresh your repository, you should see the data then there. Like Andrea mentioned, metadata is useful to keep your data meaningful. OMERO supports the following types of metadata: so-called descriptions, which are free text annotations, and are used to provide general information and context about the project or the data set. Then text can be used, especially due to the two-level hierarchy in OMERO, to help to search for the data and find it easily. You can use text to say, for example, this is experiment series A, experiment series B, and so on. These are just simple labels which are added to the images. And then there are so-called key value pair. This is a form of structured metadata. By the way, I can show it here to you. This is the description, short description of the data set. Here you have the text. These are just some short labels which are added to the images. Then there are the so-called key value pairs, where you have a certain key entry and which can have then various types, like, for example, experiments. You can say then A, B, C, and so on. And then you can also upload attachments. This can be PDFs, simple files, but also small images to help you to, for example, upload how the experiment, how the preparation of the experiment was done. Or another form of metadata are so-called comments, where these are also free text annotations, which gives, for example, some add notes about the observations which were done or how the experiment went. And two other forms are so-called ratings, where you can give stars to images, are also called tables. The metadata can be used to also to search for certain images, whereby there's so-called simple search form, where you can search for certain names or also for certain text. However, it's also possible to search for certain key values, but using this search syntax. OMERO also comes with a so-called advanced search form, where various simple search entries can be combined with so-called Boolean operators. Yeah. In order that OMERO users can collaborate with each other or share their data, they have to come together in groups, and these groups know two different types of roles. There are so-called simple group members, which can collaborate with each other, and so-called group owners, which are this more or less the administrators of the group and have more high permission level than the simple group members. There are 4 different forms of group types, how the group members can cooperate with each other. That's the so-called private group type. It is actually not really a group type at all. It's more used for, let's say, a professor who has to manage several students which do not need to know from each other and cannot see the data from the other persons. Then there are so-called read-only, is the read-only group type, where the various members of the group can see the data but cannot manipulate or add additional information to the data. And then there's the so-called read-annotate type of collaboration, where the group members can annotate the images and so on, add interesting regions of interest, comments, and so on. This is preferred if you are working with, for example, an external collaborator on a common publication. And then there's the read-write type, which is the last type and is used for collaboration of people who trust each other and have full access and rights to the data of each other. So this is how the groups are defined. It's very easy. It's just clicking and defining the group owners, group members, and read-write. And if you save it, a new group is established. In order to give you to dive a little bit deeper into this, I want to give you a short overview how the group owners, the actions group owners can perform on the data of the group members, and the group members can perform on which actions group members can perform on the data of other group members. So if you see here private, a group member can manage data of other group members, cannot change ownership, even doesn't see or can view the data, like I said before already, and so on. And here you see read-only, yes, can only see, like I said before, read, annotate, can just annotate the data. And here by read-write, it is possible to see, view the data, annotate the data, edit, delete the data, copy and move the data. And the group owners, as you can see, have, except for private, nearly all the rights to do all the actions, even change the ownership of the member data. Yeah. So before I start with a live demonstration of OMERO, I want to highlight that there are OMERO courses taking place at the Medical University of Graz. This 3-hour introductions of OMERO points out the core concepts and strategies, how to organize your data, and also possible pitfalls for working with OMERO. And if you're interested, you can book it over the Medical Research Academy. And yeah, next one is starting soon, approximately 2 weeks. So this was it with the presentation so far, and I will now log into OMERO. And when I open OMERO, I will see my standard user repository, like I said before. And yeah, if I open, here is my repository view. Here are the images of my data set, and here is the information area. Exactly. So if here, this part helps me to organize my data, to create, for example, a new project or new screens. I can then copy and paste the data and so on. This part here gives me information about the images, shows here, for example, the texts which are used, and also, for example, there are useful links then to preparation protocols stored in the eLab, which is accessible directly by clicking. Also, I can then show and download then various metadata attached to the image. This area here helps me to get different information about where the file is stored on the server or where it was imported from. Here it is possible to download the image in various file formats. I can also see, for example, then here, if I use a different image, get information about the acquisition of the image. For example, the type of microscope that was used and the magnification factor and a lot of other different stuff, and also about the channels which were used for the acquisition. Yeah. I can then have a look into the image. If I double click it, the OMERO viewer opens, which allows me to zoom into the image and take a look into it, to also manipulate the way the data is viewed, because normally the software which comes with a certain microscope or file format is optimized for this file format and for this microscope. This isn't the case for OMERO, so you have to do a little bit of optimization of the images. However, this can be done just for one image or for all the images in a certain data set. So you can optimize it also using the histogram view. It's also possible then to optimize the various channels. You can add a different color for a channel, for example. And after you have done all this optimization, you can save it either for this particular image, or you can apply the changes, the optimization changes, to all the images in this folder. It is also possible to show or to add regions of interest to this image. This is just an example where I did some training already on this image, but it's very simple to add a new region of interest. You just select here the form, how the region of interest should look like, and then click twice and can then add a comment, for example, useful area, and okay, and save it, and then a new region of interest is created. It is also possible to change this region of interest, give this region a different frame or even a different border, for example. Form you can add errors and so on. And once this is done, you can then save it, and then this new region of interest is uploaded then to OMERO. Yeah, exactly. So that's mostly it. I can also then delete regions of interest which haven't been that promising, for example. And so this is just then queued for deletion. If I save this state, then these regions of interest are then removed. In order to create, to upload new images into OMERO, I will show you how to use the OMERO Insight desktop client. So it starts up and shows me on the left my repository. I can then just say upload new images. It opens up then with the view of my local directory, and I can select here some example images, which are small TIFFs. Just select them for import, either use an already existing project or create a new project. I can say specify here the group I want to import the data to, and also I can say if I'm an administrator, I can import the data on behalf of someone, which is useful for the members of our core facilities. So in this case, I'm going to create a new project. And as well as a new data set, which I call just test, create it, and then add the data to the queue. I then can here, for example, change the import options. Or if I do not want to import this data, just remove it. Here's also the possibility to specify our metadata, which hasn't been specified. These are some templates in order to fill in some technical metadata for import. However, if everything is done, then just click the import button, and then the import process starts, consisting of the upload as well as of the processing of the images. If I click then close, I then have to renew the view of my repository, and then the new project folder appears containing the images. The OMERO Insight client also comes with a viewer and other functionalities like some management tools as well. However, it is considered to use the Insight client just for small tasks and do the rest because of the better programming in the OMERO web client. So I then can quit the OMERO Insight and go back to my OMERO web. if here renew the view as well, then the imported data appears as well here. So what else is to say? For example, here I can see history. It gives me also helps me to see when data was imported. There is also the possibility to see, for example, which tags are already used. Yeah, in order to help me to structure my data and make it searchable. Here I have the possibility to run various scripts for analysis, or also, which is very useful for annotation purposes, to upload key value pairs in order to keep the data meaningful. Yeah. So more or less, this was an overview of OMERO. I hope it was useful for you, and yes, we'll be glad to, if you have any questions, to help you to answer them. So thank you very much for your attention.